Hello. Let's try to complete our studies of digestive system anatomy and physiology. This is part 4, and I believe that residual material that we have to cover is going to be just a good fit for this recording. If there would be a need, we can expand it to part 5, but somehow I'm still confident this might be part 4 being the last one in the digestive system tract. So let's go and let's share the screen. Today we're starting from anatomy of the small intestine because with the stomach we're done, and practically that is something that you and I have completed in a part 3. So part 4 practically starts at this point. Once the chyme has emerged from the stomach, basically we know it carries significant acidity and number of different active chemical compounds that were released by the stomach. The chyme enters at pyloric sphincter into the first part of the small intestine, which is named the duodenum. If its name is translated, basically duodenum means 12 width of our thumb, so it's shortest section of the small intestine. It's about 20-25 centimeter long in total. And interestingly enough, it is pretty much retroperitoneal as opposed to other parts of the small intestine that are intraperitoneal and supported and suspended by mesentery from the posterior abdominal wall. Within the duodenum, we're going to spend a little bit of time because this is the interesting part of the digestive tube, as both the pancreas with its main and accessory ducts, as well as common biliary duct, bringing bile from a gallbladder and directly from liver, is finding its way into duodenal cavity where chyme becomes mixed now with the bile as well as with pancreatic secretion. This is something that I'm sure you already understand because the pancreatic secretion is pretty much rich in bicarbonate ions, immediately brings the acidity in a more favorable position. That excessive acidity coming out of the stomach is now slowly converted to pH values that are tolerant by the small intestine and its mucous membrane, which doesn't have the same mucus protection coat as what the stomach had. Should our journey continue from duodenum, we're getting into the second part of the small intestine, which is known as the jejunum. As a rule of the thumb, most of the jejunum is located in the upper left quadrant of the abdomen, and then seamlessly without any anatomical landmarks that would be easy for us to recognize, basically jejunum becomes ileum, and again, as a rule of the thumb, most of the ileum is located in the lower right quadrant of the abdominal space. Overall, the small intestine appears to be the longest segment of the digestive tube, practically being at least 70 to 80% of the entire length. And what is very important functionally is that through the walls of the small intestine, basically most of the nutrients are absorbed either into bloodstream or into tiny lymphatics that are also part of the intestinal mucosa. We're going to show this a little bit later. So for now, we just wanted to see what it looks like. And on the next slide, you will see something that is truly unbelievable. This is the longitudinal section through part of the jejunum where you see the lumen and this kind of really numerous rich folds that the mucous membrane throws itself into. These individual folds are known as a plicae, circular plicae, plica circularis, and the main reason for their existence is that they increase significantly available absorptive surface of the digestive system. Without going into details about how do we calculate this, but should you think about just the circumference of the small intestine being, let's say, about 10 centimeters, and let's make it 600 centimeters long or 6 meters. Basically, when we would multiply these two numbers, we would get this area of basically less than 1 square meter of absorptive surface. When we add plicae circularis and then we add villi that are submicroscopic projections that are seen on the surface of plicae, we're easily coming up to absorptive surface of the small intestine being something comparable to over 200 square meters. So from less than a meter with plicae and villi, basically we're having unbelievable expansion of this really intensely valuable surface. It is the jejunum which has way more circular plicae because it is closer to stomach. It is the first part of the small intestine next to duodenum. And certainly the jejunum is bearing the brunt of all the nutrients that are still present in the chyme, which jejunum has to start quickly absorbing. As the plicae reduce a little bit and their numbers are less intense, basically we're stepping into territory of ileum, which is just looking a little bit more pale. Surface of the jejunum is more brighter red in a living person due to more intense vascular supply, which again supports more intense process of absorbing the nutrients. A little bit less blood comes into a wall of ileum. It doesn't mean it's significant. It just looks a little bit more pale. And on the top of that, reduced number of circular plicae. Within the wall of ileum, we're seeing more frequent presence of mucosa-associated lymphatic tissue or MALT. So that is what you and I can use to practically define through prism of anatomy what the small intestine looks like. And now we can move on to see what is its mucous membrane shaped as. It will remind you to some extent to what you and I have seen earlier when we were talking about different cells and different parts of mucosa of the stomach. But keep in mind, stomach had deep pits, whereas obviously those finger-like projections appear to be one of the most direct observed differences between mucous membrane of the stomach versus membrane of the gut. Basically, these finger-like projections are known as villi. Singular is vilus. And if we cut across a single vilus, we're seeing presence of smaller caliber, arterial and venule, and of course capillaries. And in the middle of them, there is something that perhaps you did not expect to see. There are lymphatic vessels that are called lacteals. So let's explain right away what's happening here. Across the epithelial surface of the villi, which has additional projections on its apical part being called microvilli, basically absorption of nutrients starts taking place. As the nutrients are absorbed, they're basically carried across the wall of mucous membrane, across the epithelium, directly into the bloodstream. This only applies to carbohydrates, sugars, and amino acids that are absorbed directly into the bloodstream. When it comes to lipids, we are terribly concerned about lipids clustering together. And that's why they are not allowed to have direct entry point into circulatory system. Their absorption is going to be done a little differently. And after they're absorbed, basically they're going to be absorbed into lacteals. So lipids into lacteals, while carbohydrates and amino acids could go directly into bloodstream. On the far right side of the screen, you see that we still have four well-known layers to us: mucosa, submucosa, muscularis, and serosa, or we can now say that is peritoneum, because the jejunum and ileum are practically entirely intraperitoneal. Two layers of muscles, circular and longitudinal, plenty of blood vessels in the submucosa, glands, and also pay attention that we have cells that are forming the epithelium being somewhat different colored than just this predominantly lighter or slightly darker shade of pink. Let's find out what cells of the small intestine look like. Very similar type of exercises what we have done for 5 different types of cells in the mucous membrane of the stomach. Let's address first the most abundant ones. They are absorptive cells, basically cuboidal to columnar in shape with lots of microvilli, so microscopically visible hair-like projections on their apical surface. Microvilli are that infamous brush border, and as we speak about brush border enzymes, basically these enzymes are present here, splitting larger molecules into smaller ones that are absorbable. So that is why we have abundance of cells showing presence of microvilli on their surface. Scattered in between them here and there, we have a couple of goblet cells secreting mucus because regardless of pH value of a chyme, basically a little bit of a mucus always comes handy to protect whether it's slightly alkaline or slightly more acidic. We need to be aware of physical protection of the lumen of the gut. Then those turquoise blue colored enteroendocrine cells, obviously enteros gut and endocrine cells inside the lumen of the gut, secreting hormones such as secretin, cholecystokinin, or GIP, gastric inhibiting peptide. And finally, at the lowest part of the screen, you have some yellow colored cells that are called the Paneth cells. They secrete lysozyme, which is bactericidal, and these cells are also capable of phagocytosis. So obviously it's all geared towards different functions, such as what the small intestine has, that is absorption, and a little bit about its own self-protection. And the key point here is to recognize presence of villi and, of course, surface of villi covered mostly with epithelial cells exhibiting microvilli on their apical surface. This is one 45 times magnification picture of what mucous membrane and other associated submucosa and muscularis layer of the gut would look like. So very good indicator that previous slide actually quite accurately reflected what's happening inside the gut. And then after the small intestine, I believe that it would be the most logical thing for us to proceed and to say a couple of words about anatomy of the large intestine. So you could have seen it on the first slide that we have used today, and I'm just going to quickly scroll back to point out that basically the large intestine practically goes and almost makes a framework within which the coils of the small intestine are placed and located. And of course, the large intestine, its large attribute is not really based on its length, but rather on the circumference or caliber. And very clearly, you can see that this is looking totally, totally different compared to anatomy of the small intestine. So as we start describing and defining what the large intestine is, basically we have to first look for the terminal part of the ileum, which comes and directly connects here to the first part of the large intestine, which is called the cecum. They form what is called ileocecal junction, and at the point where ileum and the cecum connect, basically mucous membrane creates a bit of a loose fold, and that fold is acting as functional valve that is preventing that the chyme re-enters back to ileum from the cecum. So it's more a functional valve. This word sphincter really should not be here because there is no circular arrangement of muscle fibers around this area. Cecum is interesting for another thing for us because at its base, another interesting and important landmark is what we see. It's the vermiform appendix. As you can see here where I'm pointing with a cursor. The appendix is also internally hollow. It has its lumen, and its walls are loaded with mucosa-associated lymphatic tissue. Certainly, there is a large number of bacteria, which are saprophytic organisms in the large intestine, and eventually they could be at fault for creating a situation where the appendix becomes irritated and inflamed, generating an appendicitis as a condition for which the vermiform appendix sometimes have to be surgically removed. Vermiform appendix does have its own mesentery that is illustrated here as the mesoappendix, but the rest of the large intestine, the cecum, basically becomes again retroperitoneal. From cecum, you and I go up on the right side of the abdomen, following the outline of ascending colon, which comes close to the underside of the liver, making a right colic flexure or hepatic flexure, and turning itself into transverse colon. The transverse colon, again, is interesting for us because it's intraperitoneal part of the large intestine, running across from right to left, and once it reaches the region of spleen, it makes another turn, left colic flexure or splenic flexure, and transverse colon becomes now descending colon, which is again retroperitoneal structure. As the descending colon descends down towards left side of the false pelvis, basically it is forming a section that looks like the letter S that is called the sigmoid colon, as the sigmoid colon practically brings the large intestine back into midline. Followed after a sigmoid colon comes the rectum, which sits anterior to the sacrum in the midline, and the rectum is now coming closer to its end, first forming the ampulla and then anal canal, and finally terminating with external opening that's called the anal opening, illustrated a little bit better on the right-hand side of the screen. Anal opening, characteristically described as with two different sphincters, external sphincter, which is part of the pelvic floor, voluntarily controlled, and internal anal sphincter, which is involuntarily controlled. So practically that is something that we acquire as the additional skill in the first couple of years of our life to have a bit more of a voluntary control over sphincters. So defecation and urination pretty much becomes then voluntarily carried act. Other than naming cecum, ascending, transverse, descending, sigmoid colon, and rectum, you can also see that there is something else which is looking totally different compared to what we have seen on the surface of the small intestine. Basically, four layers, mucosa, submucosa, muscularis, and serosa, are still present even in the large intestine. However, arrangement of the longitudinal muscle fibers in the muscularis layer practically condenses them not to go 360 degrees circumferentially around the lumen of the large intestine, but rather condenses them in the form of three longitudinally running bands. These bands are illustrated here, and they're called tinea coli. You can see here one, here's another one, and of course the tinea are now having effect of what is called kind of drawstring effect. Their contraction can significantly reduce the length of the large intestine, and this is particularly important for movement of the fecal material, which is having less and less water as it was almost entirely absorbed, and of course turning it closer to solid. It requires much more of a strength to move it along the terminal part of the large intestine. So what happens inside the large intestine due to tinea coli is the process, which is not just simple peristalsis, undulating wave of muscles contracting and relaxing, but rather we call it mass peristalsis, where a segment of the gut appears a little bit more in direct control of longitudinal muscles, shortening it, and circular muscles just producing unneeded propellant to push forward or to push more distally the fecal material. Because of Taenia coli that act as the drawstring, basically the surface of the large intestine also looks like being thrown into a recurring form of outpouches. And these outpouches are named individually as haustra. Haustrae is the plural, since the content which comes from ileum into cecum is still more liquidy. Basically, house tray are acting as some kind of dishes that get filled and then they spill over their content into the next one and so on. And of course, the role of house tray is going to be further and further reduced as the fecal material is formed and it is, as I said, being more solid than liquid. In addition to host trait, Taenia coli, one can also see on the surface of a large intestine additional adipose tissue that is arranged quite randomly in the form of epiploic appendages, and it is quite individual finding. Some people have more, some people have a little bit less. However, it is typical and standard finding on the surface of the large intestine. As we wanted to compare small intestine to stomach, now we have a chance to compare what large intestine's mucous membrane looks compared to other two. Still, we have same four layers: mucosa, submucosa, muscularis, serosa. Mucosa appears to be quite thick compared to other layers. Basically, it is having a large crypt-like indentations, and then these openings are practically illustrating the very surface of the large intestinal mucosa as being practically flat. And of course, these openings are still submicroscopical, so we do not see or experience this as these deep pits actually containing residual parts of food, something that eventually would be subject to rotting or decomposing it in any other form, shape, or manner. The large intestine has done most of the absorption in its cecum ascending and part of the transverse column. It is unfair to say the large intestine because we know as the chyme runs through the small intestine, it was first subject to a really vigorous form of sorting out nutrients and absorbing them into either lacteals or directly into bloodstream of villi. However, small intestine, regardless of its length, cannot do everything, and there is still a little bit of water to be absorbed through the lumen of large intestine, and together with water, absorption of minerals like sodium or potassium is taking place, as well as water-soluble vitamins. For that reason, you are seeing these openings that exist within the wall of a large intestine, again having lots of cells with microvilli on their surface because they still have to do the work in absorption. And there are quite a large number also of goblet cells secreting mucus, which also serves as the protection for the mucous membrane of the large intestine. At this point, you and I can eventually start thinking about what is going on. And this diagram, although it is way better for using it while describing liver and pancreas, even gallbladder and biliary ducts, we're going to stop here for just a brief moment, and I will tell you something else that is interesting and important for what we need to do. Absorption is the key issue, and of course, in the lumen of the small intestine, most of it happens. So, since we can go as far back as the oral cavity, we remember that there was something called salivary amylase, and salivary amylase started breaking down larger polysaccharide molecules like starch and only in the upper digestive tract salivary amylase was active. Moment it would come into stomach, it becomes inactivated with gastric juices. So continued digestion of carbohydrates will have to be done only with the help of pancreatic amylase, which definitely comes from pancreas, which is a very strange form of gland, mixture of exocrine gland, part of digestive tube, and of course part of pancreas being endocrine, keeping control over levels of blood sugar. Finally, as the pancreatic amylase is bringing down larger molecules of carbohydrates, they're still basically in the form of disaccharides, so brush border enzymes of villi and microvilli, of course, that are covering their surface, basically breaks down disaccharides into monosaccharides, so sugars with six carbon atoms in its molecule, and it's the monosaccharides that could be absorbed. For the proteins, basically oral cavity was of no help other than mechanical digestion, but still proteins for proteins. Only in the stomach, with the release of pepsinogen and its activation into pepsine, basically proteins are broken down either to smaller protein molecules that we can now call peptides, up to 10 amino acids. And then peptides are to be further broken down into amino acids and eventually dipeptides, so two amino acids held together, could be absorbed. So pancreatic enzymes such as trypsin, chymotrypsin are assisting in the breakdown of proteins, and dipeptidase enzymes splits dipeptides into individual amino acids that are then easiest to absorb in the small intestine. With lipids, you remember that we have a bit of an issue as lipids are not directly absorbed into bloodstream because they would be more dangerous and they would have perhaps more damaging effect if they would be allowed to freely move through the circulatory system. Digestion of lipids started already in the oral cavity. You probably remember lingual lipase, which wasn't particularly effective because it needed also activation by stomach's acidity. And also gastric lipase, produced by stomach, finally starts breaking down lipids. Once they're in a chyme, basically it will require, and that is one of the reasons for which this diagram is really the best and optimal, because I can now point to you directly here. This is the biliary tree, and this is its entry point into duodenum. So with the bile, which is required for emulsification of lipids into smaller particles, basically mixture of chyme with the bile is a must to do for any of the lipid molecules to be absorbed. And then pancreatic lipase, another enzyme that helps us break down the lipid molecules. Pancreatic lipase is going to split triglyceride. So if you remember what triglyceride molecule looks like, it's glycerol to which three fatty acids are attached. Basically, pancreatic lipase leaves two fatty acids free and a monoglyceride, which is one fatty acid still attached to the glycerol. And these fatty acids, individual or monoglyceride, can be absorbed across the wall of the small intestine into lacteals. Regulation of intestinal secretion is mostly done through locally elicited enteric reflexes. That is something for which I don't have enough words to say how important it is and how important it is not to bother, so to say, the brain with its additional activity, thinking about, okay, what did you consume? Is there more proteins or more carbohydrates? How do we set the proper digestive mechanism? It is all done locally through locally elicited reflexes. Stretching of the wall, whether it's a stomach, whether it's chyme entering into small intestine, is one of the most important physical things that mechanoreceptors detect. And of course, the different composition of chyme basically triggers different secretions and different form of enzymes, as we have just added them in the past few minutes. Chyme, as it travels through the small intestine, it doesn't go at any high speed. It's about 10 centimeters per second. So it's going to take about 4 hours that are required for chyme to clear the small intestine. Now, a little bit about absorption. And for the absorption, I would prefer if you and I can just go to this diagram, and don't worry, we'll come back to liver and pancreas later on. So just to make sure that we understand what's going on. Absorption is the key thing because they are locally absorbed ingredients that the body will either use to break them down to form the energy, adenosine triphosphate, or some of the absorbed particles could be used for creative, constructive processes as the body is rebuilding itself. Glucose, galactose, and fructose on the top of the screen are 3 monosaccharides that we consume pretty much with regular diet and food intake. As you can see, glucose and galactose are going to have secondary active transport together with sodium ions to get into cells of the small intestine, while the fructose, due to its slightly different shape of a molecule, will require facilitated diffusion in order to penetrate through brush border and move further from cells that absorb it into bloodstream. You recognize that we needed those brush border enzymes to split disaccharides into monosaccharides. So brush border enzymes are in this area. Absorption of amino acids, dipeptides, and eventually tripeptides. 95 to 98% of that is absorbed in the small intestine, and most of the amino acids are absorbed through active transport or secondary active transport along with the sodium ions. However, dipeptides and tripeptides use the secondary active transport together with hydrogen ions. Once they have been absorbed, basically amino acids could freely diffuse out of the cell. And keep in mind that monosugars would go and diffuse, but it's facilitated diffusion into bloodstream. And finally, lipids, those ones that are interesting for us as they cannot be absorbed directly, we actually have them diffusing through simple diffusion, high to low, if we're talking about short chain fatty acids. Because as we know, cellular membrane of pretty much all living cells is a phospholipid bilayer, so fatty acids are pretty much hydrophobic as much as the cellular membrane is, so they blend nicely together, and that's why simple diffusion is marked here for short chain fatty acids. They diffuse into cell, but they also diffuse out of the cell, but this time they could go further out. For fatty acids of triglycerides, basically we know that we first have to use the pancreatic lipase to split them into long-chain fatty acids, 2 of them, and the third one is still attached to glycerol, making it monoglyceride. That's when we need bile in order to form so-called micelles. Basically, micelles are bile salts that form a loop or a circle around individual lipid molecules, practically making the center still being hydrophobic, while the bile salts make the entire micelle being hydrophilic. And that is, of course, why we need to insert and add bile in order to see complete absorption of lipid molecules. So micelles, whether they contain longer chain fatty acids or monoglycerides, are still fully compatible, and they simply diffuse into a cell across the microvilli brush border. And within the cell, something interesting happens. Basically, fatty acids recombine with the monoglyceride and they form the triglyceride. However, the triglyceride is going to be now again modified into what is called the chylomicron, because within the cell, synthesis of proteins that is binding to lipid molecules is a must, because the proteins are now acting as the carrier molecules. So with chylomicron basically being formed within the cell, it will be sent out of the cell into a lacteal of a pilus. Lacteals will start merging, getting bigger, and ultimately we're going to have all the lymph collected from the entire length of the small and large intestine into cisterna chyli, that is pretty much the largest accumulation of lymph within the body. And from there, from cisterna chyli, basically that's the origin of thoracic duct, which carries those chylomicron structures into junction between thoracic duct and circular cardiovascular system, that is at the junction point between internal jugular vein and subclavian vein. Together with lipids, we are also absorbing in the same form the lipid-soluble vitamins, vitamin A, vitamin D, vitamin E, and vitamin K through the walls of a small intestine. In addition to carbohydrates, proteins, lipids, basically something else is going to be absorbed. Almost all of the sodium is absorbed inside the small intestine, most of it through sodium-potassium pump. Also, bicarbonate, chlorine, iodine, they follow sodium, or they are actively transported from a lumen into a cell, including, of course, calcium ions, iron, potassium, magnesium, phosphate ions. They are absorbed using active transport. So calcium, iron, potassium, magnesium, phosphate, active transport. Vitamins B and C, which are not really liposoluble vitamins, they're absorbed through simple diffusion. We're also expecting vitamin B12 to combine with intrinsic factor that is produced by parietal cells of the stomach, and then it becomes absorbed within the ileum. And finally, what is the most important ingredient of our life? It's the water. So we're talking roughly about 10 liters of liquid secretion or ingestion that is entering our digestive tube on any given day. Basically, we're going to have water, majority of it, practically over 80%, 90%, being absorbed in the small intestine, and only residual water is going to be absorbed by the large intestine. So I'm just going to slow down here a little bit and bring us back to make a little summary of what happened so far. So we talked about anatomy of small and large intestine, basically talking about duodenum, jejunum, ileum, and then different parts of the large intestine. We also compare the mucous membrane of both small and large intestine, and we compared it to what we have seen in the stomach. And as a result of that, basically we were able to recap different type of enzymes. And in the last section, you and I were talking about different form of mechanisms being used for absorption of carbohydrates, proteins, lipids, but also including lipid-soluble vitamins, waters, and minerals. So we're to practically stop here because I really don't think that we're going to have much of a space for both liver and pancreas. Otherwise, this recording could be a bit too long. I believe that we can just add something about large intestine and what's happening within it so that we complete both small, large intestine as digestive tube, and we are unfortunately to leave as a separate recording anatomy and physiology of liver and pancreas. So let's go back to one of the slides that is covering at least what we need to know about large intestine. So just give me a chance to go back and scroll through this. We're going to start from this slide, and I need to go a couple of slides back until we get to this point. One of the most important things inside the large intestine is bacterial flora. So we're talking about 700-ish different species of bacteria, and they are non-pathogenic, so we really should not have any fear of them. They're basically symbiotic organisms that are causing no harm to us for as long as their colonies are controlled and they stay inside the lumen of the gut. Basically, bacteria are going to be quite helpful, and we need their presence. So it is something that we normally expect large intestine to have. Many of these bacteria will facilitate chemical digestion and absorption. Some of them will synthesize certain vitamins, mainly biotin, pantothenic acid, and K vitamin. Some of the bacteria are linked to increased immune response. Refined system prevents these bacteria from crossing the mucous membrane barrier. So that is something that is definitely no-go for them. First, there is peptidoglycan, a component of bacterial cell wall, which immediately, upon its detection, activates the release of chemicals by mucosa of the large intestine, drafting immune response. Especially important for that are dendritic cells in the mucous membrane of the large intestine. Dendritic cells open the tight junctions between epithelial cells and extend their probes into the lumen to evaluate different microorganisms and their antigens. Those dendritic cells with antigens then travel to neighboring lymphoid follicles in mucosa associated with lymphatic tissue. T cells are now presented the antigen, and through this process basically triggers release of immunoglobulin A mediated response. So the bacterial flora is definitely held in constant check. So for the residual part of absorption, which needs to take place across the wall of the large intestine, basically we're thinking about minimal amount of chyme, which still contains some nutrients, and the water. Basically, chyme stays within the large intestine anywhere between 12 to 24 hours. Ileocecal valve or ileocecal mechanism is what prevents chyme from returning back from large into small intestine. And we're going to have also some kind of interesting reflexes, which essentially are triggering movement of material through the large intestine. One of them is gastrocolic reflex, basically upon any kind of ingestion of food, particularly in the morning, with the first cup of tea or a cup of coffee or a couple of bites that people like to have upon waking up. Basically, the stretch of the stomach sends a very strong reflex for large intestine to increase its mass peristalsis, and that is pretty much to move the remaining material that is turning into feces closer to its elimination point. Chemical digestion in the large intestine, basically, although the glands of the large intestine are secreting mucus, they do not secrete digestive enzymes. So no enzymatic activity. Basically, chemical digestion is exclusively done because of bacteria that reside in the lumen of the colon. Through the process of fermentation, bacteria break down eventually some of the remaining carbohydrates. Keep in mind there should be none of them, or at least a very minimal amount. And as a result of this fermentation, discharge of hydrogen, carbon dioxide, and methane inside the large intestine is happening, which result in the production of gas, which from time to time also needs to be eliminated. Absorption of water is also part of the large intestine's role. We're talking about slightly less than 1 liter of fluid that will be absorbed, and some sodium, some chloride, and of course, the water-soluble vitamins. And finally, we need to address something that is movement of the fecal material into I'm sorry, because at this point, I'm just not sure whether recording is still running. It is. I'm sorry, because I thought that I press accidentally pause, and then I thought that I need to do this all from scratch. Forgive me about this. So we're talking about defecation reflex. So we have it here on this slide that fecal material accumulate within the ampulla of the large intestine. And once again, sorry for this back and forth through the slides, but I just wanted to have something that is at least on the screen. So as the fecal material fills up the ampulla of the rectum, it starts slowly stretching out and distending its walls. When it reaches the critical mass of around 150 milliliters to perhaps 200 milliliters volume, it is going to stretch the walls to that critical threshold point. So sensory fibers that are part of the rectal ampulla will run through S2, S3, S4 nerves into spinal cord, informing the sacral segments of the cord about stretch being reached to that critical point. And the same S2, S3, S4 fibers are going to send motor signals back to large intestine, basically to increase contraction of longitudinal rectal muscles, and that event is going to lead to the process of eliminating the fecal material or defecation. There are some accessory mechanisms such as compression of the abdominal wall that is also required to increase the intra-abdominal pressure, as well as pretty much control of sphincters. As we know, internal anal sphincter is involuntarily controlled, and external anal sphincter with its voluntary control will have to be relaxed in order to allow for the defecation. So at this point, I believe that you and I are done with part 4. I was kind of over-optimistic for making it just one recording today. Looks like we still have to do the 5th one, liver and pancreas and bile. And I'm going to gladly do it perhaps today, tomorrow, the moment I have some extra time to complete the digestive tube. So wait for part one. And if you like part four, I'm very happy. Stay well. See you soon. Until then, take care and bye bye.